The overall goal of this procedure is to seed, expand, and differentiate neural stem cells that were prepared from the hippocampus of adult non-hibernating yearling Arctic ground squirrels. This is accomplished by first seeding the AGS neural stem cells. Next, expand and seed the neural stem cells into differentiation medium. The third step of the procedure is to differentiate and maintain the neurons. And finally, the cells are fixed or treated and fixed for analysis. Ultimately, results can be obtained that show AGS neuronal progenitors continue to divide through 2 weeks of differentiation maintenance and even under ischemic conditions. Neuron numbers are determined by staining and fluorescent microscopy techniques that measure neuron ratios.
The implications of this technique extend toward the discovery of therapies for stroke and cardiac arrest, since Arctic ground squirrel differentiated neurons are tolerant to all ischemic insults.
Prior to starting this procedure, prepare the flasks to be used in AGS-NSC expansion by coating them with polyl-ornithine. Thaw the polyl-ornithine working solution by leaving it in the incubator. Once thawed, the solution could be stored for up to 1 month at 2 to 8 degrees Celsius. Add 8 milliliters of polyl-ornithine working solution to a vented T75 flask and evenly distribute the solution over the entire culture surface of the flask. Incubate the flask at 37 degrees Celsius for a minimum of 24 hours, preferably in an incubator without added CO2. Prior to use, aspirate the polyl-ornithine working solution from the flask and wash twice with sterile tissue culture grade water. Aspirate the liquid until the flask is nearly dry before use. To thaw and